Hopefully we save the best for last. Today's session is on immunological data for decision making. So we have an interesting lineup of speakers, and we're going to hear some interesting data around pneumococcal vaccines and their immunogenicity, and it's a good follow-on from the talks we've heard today already. So I think it should be a nice way to finish today's session. So yeah, on the next speaker today is we have the pleasure of Dr. Tuan Nguyen, who's a medical doctor. We heard from him earlier with public health, epidemiology and vaccinology background, and he's the head of the Clinical Research Centre at the Pasteur Institute in Ho Chi Minh City. Has extensive experience in clinical trials and evaluation of health programs, and also provides technical support to members of the National Immunisation Program in Vietnam. So welcome again, Tuan. Thank you.
Thank you, Paul. Good afternoon, everyone. First of all, I would like to thank the organizer and Paul and Kim who invited me to be here. It's the first time in Melbourne. So after the statistical training section, it's very interesting. For my turn, it's a little bit easier because I think from yesterday until today, you already hear something about the data from Vietnam regarding the immunogenicity and carriage from the different schedule. Today, I would like to show the data come from one of the complex clinical trials in Vietnam. To compare the immunogenicity of different PCV 10 scheduling, and also compare with the PCV 13. We focus here in the immunogenicity of the alternative 10 valent pneumococcal conjugate vaccine for scheduling. It means the GSK vaccine in infant in Ho Chi Minh City, one of the biggest cities in Vietnam. It is RCT, randomized control trial, but the single blind because of the structure of the vaccine. We have the open label, but still parallel group and randomized. My presentation will be focused in 4 parts: the background, the methodology, some finding, and some conclusion. So before this study, what we know about the BCV, I just pull around the question why we have the PCV vaccine for infants in more than 10 years ago, but now the vaccine coverage is not quite high. If we compare to the COVID-19, just 1 or 2 years, we covered all of most of the country, but for PCV and infant, because the high burden of the PCV. Until now, we have 43% of children globally do not have access to the PCV vaccine because they live in a country that have not yet introduced PCV, including my country also. Even we have the vaccine, but it's just in the private market. And the appropriate and optimal PCV schedule for low middle income country is still lacking. We have something, but it looks like not enough to persuade the government or the policymaker to make the decision. And we have, based on the previous study result in South Africa and Nepal, we already know that the primary schedule 3 dose gives the IgG response higher than 2 dose, and the longer interval is higher antibody response. But unfortunately, a little is known for the effect of PCV dosing schedule on immunogenicity and nasopharyngeal carriage in low middle income country in Asia. And that's why at that time, Kim and his team come to Vietnam, and we discussed to set up a randomized control trial in 2 urban districts of Ho Chi Minh City. Ho Chi Minh City is one of the biggest cities in Vietnam. with more than 10 million people there. And it's located in the southern of Vietnam. And at that time, we still not have the PCV in the market because the vaccine have to do some preclinical study before licensing. We come with the research question is two-dose primary series of PCV is non-inferior to three-dose primary in terms of immunogenicity. And I can say when I approach this study, this is one of the complex study design in vaccine trial. It's not only the number of subjects, but also the different schedule and different outcome. I show you here is the main study. The main study is we include around 1,200 subjects, and we random the subject, the infant, we enroll them at 2 months of age. We enroll them in 6 groups. We call the group A, B, C, D. Is use the PCV10, and for the group E, it's BCV11, and we have the control group. Regarding the number of subjects who are randomized, we can say that the ratio is 3, 3, 5, 4, 5, 4, and we random by block in 2 districts. And because of the subject is enrolled at 2 months of age, we not only give them the study vaccine, but also the NIB vaccine because it based on our requirement. And we give them the co-administration vaccine with hexavalent, DTP, polio, HIP, hepatitis B, and also the measles at the 9 months of age. And it is the schedule for BCV10. We have the group A is 3+1. That means it follow exactly with the recommend of the company. 3 dose at 2, 3, 4 months, and the booster at 9 months. 3+0 at group B, no booster. 3+1, the interval is 2 months, and the booster at 9 months also. And the group D, just 1+1 at 2 months and 6 months. That means the interval is longer than the group C. And for the blood sampling, to evaluate the immunogenicity, we take the blood at a different time point, but you can say that we take the blood sampling at the 1 month after primary scheduling. If they complete primary, we take the blood to do the immunogenicity evaluation. And then at the time frame is 6 months, 9 months, 1 month after booster, and at the 18 months. We not collect the sample all of the subject. We just take in some blood sampling visit, we just take half of the subject just to reduce the risk to collect the blood in uniform. We divide compared to the group and the subject, and we do the assay with ELISA and optional Fraser society. And here's some result. I'm sorry, it's not. I'm not sure it's not clear enough for you, but I try to show some data regarding the first of all regarding the comparison of 2 dose with 3 dose with 2 dose after primary vaccination 1 month. And here the immunogenicity with the criteria is IgG 0.35 or higher. And you can see here, if we compare the 3 dose, that means the group A and B. Group A is 3+1, group B is 3+0. We compare with the group C, that means just 2 dose, 2+1. And for all of the serotype in PCV10, we see the proportion is of participants have reached target, the threshold is similar in group. It's a bit higher in group A and B. That means the 3 dose. If you see in the GMC data here, but totally the group C is not non-inferior than the group A and B. It for the immunogenicity, if we're looking for the IgG. And now if we looking for the functional antibody, it's a little bit clearer if we see the 3 dose and the 2 dose, especially for the GMC. It's a bit higher than group C. Just for some serotype, it's not clear enough, like the serotype 4 and 19F. So for 3 and 2 dose, after primary schedule, the immunogenicity, if we're looking for the IgG in general or the functional antibody, the 2 dose is not really non-inferior. Even the concentration of 3 dose higher than the 2 dose. Now we compare the 2 dose together, but different interval. 2 dose with the 2 months apart, and 2 dose with 4 months apart, also in the primary schedule. And very interesting, we see that with the longer interval, I mean in the group D, the group D is the 1 dose at 2 months and the second dose at 6 months, 4 months apart. The immunogenicity response is higher than 2 dose, but just 2 months apart at 2 months and 4 months. You can see even in IgG or GMC. It's the primary. And it's similar if we compare, we're looking for the functional antibody. The 2 dose with the 4 months apart, it response higher than 2 dose with the 2 months apart. And now we're looking for another issue regarding the GMC response at 6 months of age, when the infant complete the primary schedule. In the group that is just complete the primary dose, the primary schedule just with the 1 dose, 2 dose, or 3 dose. And the GMC is higher in the group of 3 dose compared to 2 dose and 1 dose, except the serotype 19F. That is the evaluation at the 6 months of age. So what happens if we follow up until the booster dose? And I will go this one first. So this is the immunogenicity regarding the IgG reach the target. We follow until 18 months. There are 2 things we can see here. The first one is after the primary series, for all schedule, at the time frame at 9 or 10 months, that means before we give the booster dose, more than 80% of participants still reach IgG concentration. But as I think that you concerned yesterday, it still decrease after that at the 18 months. And very interesting here, if you're looking for the blue one, that means the 2 plus 1 booster at 9 months. You compare to the red line without booster dose, or the green one, just 2 and 6 months. And you see, if we give the booster at 9 months, we will get the benefit for the second year. That means if we just give the primary schedule, maybe 3 dose is higher than 2 and then 1. But then without the booster, the second year, we still need the booster if we would like to keep the higher titer of antibody. So this slide tells us that the booster at 9 months of age, it may be useful for the second year of life of children. And here is some data regarding the booster or no booster. We compare the group of the group A and group A and here group C. Group A and group C with the booster, and we compare with the group B without booster. You see the GMC it without booster is lower than the group with the booster. And if you look at the serotype 1, it's very low. So maybe that is one of the key point to recommend. We need the booster dose in the country where we just use the schedule 3+0, and where the country the serotype 1 is circulate there. We still need the booster to increase the immunogenicity. And we also do the cross protection for other trend without in the BCV10, and we still see the response for it. And it might be the extra benefit for the BCV10 to protect in the country where we have circulate the 6A or 19.8 in the country. The last slide, we would like to show you another thing about the dose of the booster dose, and also the serotype response for 14 and 19F is not changed much. First of all, if you see here, we say the peak. Sorry, this one. You see the peak, the two peak at 7 months and 10 months. That means right after we give the booster dose at 9 months, or we give the second dose with the schedule 1+1, the immunogenicity increase, and we still protect till good until the next year, 18 months. And the big appear just 1 month after the final dose of primary in the 2-dose schedule, or at the 2+1 schedule. That means 2, 4, and 9 months. And if we compare the 3+0, that means without booster, and 2+1 with the booster. Before booster, 3+1 is higher, except the 19F. But then if we booster 1 month later, the higher end continue, and this higher still persist until the 18 months. So I don't want to show more because it's too complex to just higher and lower and then protect and not protect. But I just say that to conclude that if we compare the 2-dose primaries, we can say it's non-inferior to a 3-dose for the primary vaccination. Although the lower antibody level for most cell time with the 2-dose than the 3-dose. Second one, with the wider interval, that means 4 months apart between first and second dose, result in high antibody level than the 2-dose just 2 months apart in the primary vaccination. It's similar after the booster vaccination. Third thing, 2-dose primaries plus 4-month intervals seem to provide similar or better than protection after the primary vaccination series than 3-dose, and enable a third dose to give a booster with prolonged protection into the second year of life. But we see the blue line in the figure. So this may be the beneficial in the high burden setting when we give one booster dose, and we will have the benefit of the next year. And in the area where serotype 1 is circulate, maybe we need the booster dose. It will be support and put in the schedule 2+1 if you use the total 3 dose in your country. So I stop here. I want to thank the MCRI, thank the Manzi School and Bill Gates Melinda, and even the London School to give us the data analysis for this, and also my colleague, my friend at the Clinical Research Center and Unit and Microbiology Lab of the Boston Institute of Immunology. Thank you for your attention.
Thank you, Dr. Tuan, and you both first speaker very on time. So now I would introduce Associate Professor Paul Licciardi. He's an immunologist who leads the immunology team of the new vaccines group at MCI. His expertise lies in the evaluation of vaccine immune responses, having led immunological study for several clinical trials of pneumococcal vaccination throughout the Asia Pacific region. So, Paul.
Thank you, Anna. Yes, so it's the last talk of the day. Yeah, I'll be changing tack a little bit. So we've heard a lot about PCV immunogenicity, mainly around antibody response, which are the main correlates of protection, as Yinbun were talking about. We've been doing a lot of work around novel markers, and you heard from Tan yesterday around B cells. And so as part of the two Vietnam trials, we've been doing a lot of work over the last couple of years looking at other, or trying to identify potentially other markers that might predict protection more reliably. So just to kind of recap, we know that currently the assessment of PCVs is driven by serological assays, and these are the two kind of WHO recommended methods, serotype-specific IgG by ELISA and opsonophagocytosis. So these are the two assays, and results from these two methods are required for regulatory purposes. And the correlates of protection following these, using these vaccines, are really being based on invasive disease. So we do know there's an aggregate threshold, and it's 0.35 micrograms per mil that we use and everybody uses. But there are a number of studies now, and I think it's widely recognized that these correlates do vary widely by serotype. The serotypes are all very, very different. And so there have been studies to suggest or to show that, you know, for some serotypes, you actually need a lot more antibody, and for others, you need much lower levels of antibody. And it has been shown in some cases that for settings where there's a lot of disease, so in high burden settings, that level of antibody required is a lot higher. So what we're trying to get at is understanding correlates of protection against non-invasive pneumococcal disease, and we don't really have a correlate for carriage or for mucosal disease. And I think it's widely, thought that the levels of antibody you need is going to be much greater, because you need that antibody to actually make its way to the mucosal surface. So the question is, do we need to understand the role of other markers? And we've been focusing heavily on B cells, but I'm going to kind of change tack a little bit for this talk. At the moment, there's very, very limited data for this from randomized controlled trials. As you can imagine, it's very challenging to do these kind of analyses around novel markers. So this is really just to show you the type of response that's generated following PCV. And so the image on the left is really a polysaccharide-based vaccine, and the one on the right, which is what I want you to focus on, is a conjugate vaccine. And really, you can see clearly the differences. So we've been focusing on antibody to measure these responses, and more recently B cells. But you can see here on the right that actually involves many different parts of the immune system, particularly your T cells. That you can see here, I think. Oh, maybe you can't. My pointer's not working. Okay, so you've got T cells, you've got lots of different markers on these T cells, and they interact with your B cells, which ultimately will produce antibody. And I just put this up because we are interested in the other aspects of the immune system that might be driving long-term protection. So there are some sources of evidence. It's quite limited, but there are some sources of evidence for the role of cellular immunity. And we know, obviously, vaccination produces immunological memory, and that's driven by T cells and B cells. But in terms of pneumococcal vaccines, it's actually not very clear. There's not really a lot of studies out there, and that's mainly because these vaccines are pretty effective. But some of the data comes from comparing vaccine efficacy between conjugate vaccines and polysaccharide vaccines. Of course, more recently, there's been a number of human challenge studies, and this is where you deliberately expose individuals to pneumococcal bacteria, and you can monitor in a very controlled way their immune responses over time. And that's identified a number of interesting markers. And then, of course, we do have quite a bit of data in animal models, particularly mice, but unfortunately, a lot of these don't translate well into humans. So some of that evidence around the variable efficacy, particularly that we see between the two vaccines, the conjugate vaccine and the polysaccharide vaccine, around protection against carriage and mucosal disease, particularly pneumonia. And so you would have seen Fiona present some data on this. They do vary quite differently in terms of study design, but we do know conjugate vaccines are far more effective against pneumonia and certainly protective against carriage. So that's one key difference, because what we see when we do immunogenicity studies comparing these vaccines, we've done these ourselves, that the actual antibody levels and opsonate phagocytosis activity is actually very similar. So that kind of tells us that there are other effector functions happening that's driving that protection. And as I was just having a bit of a look, I did come across this new RCT. It's called Preventing Pneumo. It's run by the Liverpool group, where they're actually going to do a human challenge, successive human challenge, using different serotypes, and then seeing how protective that is when they do that challenge post a vaccination with either a polysaccharide vaccine or a PCV. So that will be quite interesting to see how that goes. And there's been some other lines of evidence. Earlier this year, there was a paper published looking at the comparison of the PCV13 and the polysaccharide vaccine, but looking at other antibody-mediated functions. We're focusing on currently looking at antibody-mediated binding, through the antibody combining site. But there are, of course, a lot of other other antibody-mediated functions, what we call system serology. So it's through the Fc receptors that engage innate immunity. And that actually showed that PCV13 was by far and away a very much more capable of eliciting these kind of responses and maybe providing some further insights into the role of these vaccines. So we do think and believe that there's going to be a lot of other immunological factors important for driving the mucosal protection. So I won't spend too much time on this because we've heard a lot about reduced dose schedules, but as Twana was talking about, post-booster immunogenicity is very similar, despite whether you have a 2 or 3 dose primary series. And so that kind of leading the way towards dropping a dose, potentially dropping a dose in the primary series. We've also heard that the interval is very important. It's not just for pneumococcal vaccines, we've seen it for COVID vaccines and other vaccines that if you widen that interval, you actually get a much better antibody-based response. And across the 2 Vietnam trials, we've actually seen intervals go from 2 months to 4 months and then to 12 months. And the antibody response is substantially greater as you increase that gap. And as we've heard multiple times already, in the context of mature PCV programs, where herd immunity has kind of been established, vaccine type carriage has been kind of established, that these 1+1 schedules may be more important, not only to extend that protection into the 2nd year of life, but also to maintain that level of herd protection. And then potentially that first dose can also maintain individual or provide that individual protection, which is still very critical. But by and large, we still need a lot of evidence around this from these high burden settings, particularly in the Asia Pacific region. So we have heard a little bit about trial 1 and trial 2. This is kind of the study design. And as Twan was mentioning, we like to do very complicated study designs. So this is another one, and this is trial 2, which the data is kind of emerging and being analysed at the moment. So it's essentially a 5-arm study, and I'll just be focusing on the vaccinated arms. And you can see here that there are essentially 4 groups given either PCV10 or PCV13 as a 0+1, where that vaccine was given at 12 months of age, or as a 1+1, where the vaccine was given at 2 months and 12 months of age. And so we had bloods that were collected at the time of that 12-month vaccination, but also at 13 months. And also we added an extra blood at 7 days, really to understand the cellular kinetics, because generally the cells, the T cells and the B cells tend to be activated early. And for those immunological analyses, we had a subset of 50 per group. And this is kind of our approach. So this is something we've been working on for the last couple of years, and this has been driven a lot by Jeremy in the audience here. Has been doing a lot of work establishing what we call a high dimensional flow cytometry panel, and this is really just enables us to identify many different types of immune cells that might be important. And so we take these cells, they were obviously processed and stored at Pasteur, by our friends and sent to us, and we've taken those cells, we've stained them with cocktails of antibodies for different immune cells and run them on the flow cytometer. And at the moment, we can measure 15 B cell populations and 31 T cell populations. So you can imagine a very data-rich subset of information that we're going to get. So this is kind of what it looks like. So this is a visualization of what the immune cell composition looks like. We have two different panels when we do this assay. That's why we have a B cell panel on the left and a T cell panel on the right. And you can see there are lots and lots of different populations. This is really just to show that we can visualize what these cells are. And these are, this is a way of visualizing the clusters. So they cluster in their groups, and you can see that, you know, the B cells are kind of up the top there, and they're separated away from the T cells. And for the T cell panel, we can see very clearly distinctions between the various CD4 subsets, which are important for antibodies, but also the CD8 subsets at the bottom in the green. So this is just to show us that we can pick them up, we can measure them, and they're kind of appearing where they should be. But what I'm going to try and do is switch from the B cell stuff to more for the T cells, because this is quite new, and I'm just going to pick out a few of the different interesting findings that we've found, and perhaps maybe a little bit unexpected. So when we look at T cells in particular, and we've just kind of honed in on the two key populations here, and you can see on the left, the larger box here, looking at the CD4 population, and there's kind of three main populations at the top there. You can see there's a CD4 called EM, effector memory, or central memory, CM, or TEMRAs, which are another subset, and their equivalents for the CD8. So these are the three main, what we call memory populations that we observe in the blood. And then what we have here is a visualization of the four groups. And this is a kind of a merged data set from a number of individuals in each of these groups. And so you can get a bit of a composite image of what's going on with these different cells. And what we see here, and what we're looking for here is change in intensity of the color. And so what you can see really, and we've highlighted here in the red boxes, that the staining for the effector memory population is the key memory pool that's driving long-term protection. For CD4s in the kind of orangey brown stain there in the smaller box, but interestingly enough, a very large intense stain for CD8 memory. And this is something that perhaps was not necessarily expected when we started this. And in terms of what the data kind of looks like, I've got the three populations there, and the green ones, which I've touched on, which are the TEMRAs, these are, they don't really do a lot. They're more of like a naive phenotype, but they, like they're a fully differentiated cell. So they've gone through the process, but they're expressing a naive marker. So they don't really, we're not expecting them to do much. And you can see they don't really change from baseline, indicated here as BL, and they're all across the four groups. What we do see is for CD4s, we see a very strong increase in the effector memory population in the CD4 T cells, and particularly for after one dose of PCV10 and after two doses of PCV13. That's kind of interesting because we've heard a lot of data today about this dose effect for PCV10. And as I mentioned, CD8 memory on the right, very interesting because we actually found very significant increases actually for all these groups for both CD8 central memory in the red and the effective memories in the blues. This was quite interesting for us, and it's something that we're trying to work through. There is some evidence out there that suggests that if you have certain types of pneumococcal vaccines, but also if you have an infection, that the protective response incorporates the CD8 population. The other thing I wanted to show also is another population which is kind of interesting to us is what we call a memory regulatory T cell. So this is a kind of an interesting T cell population, generally found to be associated with regulation of responses, particularly, you know, inflammatory responses. So you can see that during infection, these cells become particularly important. They're especially important at the mucosal surface, so they tend to migrate and end up in the mucosa. So this finding here is quite interesting. We get really robust increases in these cells, particularly after one dose of PCV10 or two doses of PCV13. So again, we see this kind of differential response with the two vaccines. something that we're trying to understand what the base of that might be, but that's quite interesting. It seems to play out in some of the actual outcomes that we're seeing from these trials in terms of carriage. Again, there is some limited data around these cells. We're going to try and apply some approaches with our carriage data that's been accumulating, and just to see whether these are actually doing anything or not. But it actually seems quite interesting that these are going up in response to a vaccine that we wouldn't normally expect. And the last thing I want to touch on here is another really interesting subset of T cells. Now, these are called unconventional T cells, and they're unconventional because they're kind of more like an innate-like cell. So they're not a normal T cell like that I've talked about, a CD4 or a CD8. They express very different receptors. But the interesting information here is that for all the vaccinated groups, they were all expressing this particular marker, CD69, which is an activation marker, at much lower levels than what we found at baseline. And what that suggests to us is that these cells are particularly important in controlling infection. They, again, home to the mucosa, particularly the lung. So obviously that's quite important for pneumococcal disease. And that expression of those CD69, they're disappearing from the blood. So we believe that the loss of CD69 in the cells that we're picking up in the blood is due to those activated cells basically disappearing and moving into the lung or some other mucosal tissue. And there are two kind of main key types of unconventional T cells. We call the ones on the left called the gamma delta T cells, or the ones on the right are the mate cells. And so these are really two key ones, and we find a similar pattern for both. So that's kind of consistent and seems to suggest that they might play an important role. Certainly the first time that it's been shown following a PCV vaccination. So it'll be interesting to see how that develops. Now, we also tried to kind of do a global analysis of all the different immune cells, and obviously not all of them are on this graph, but just to kind of show what's actually going on and how the responses differ, or the profile differs, whether we compare 1 dose or 2 doses of either vaccine, or in the next slide, I'll show you the comparison directly between the 2 vaccines. And so this one here is comparing either 1 dose or 2 doses for PCV10 on the left, or PCV13 on the right. And the dotted line in red is really just showing no difference. That's a line of no difference. And this is a difference in the median proportion of these cells in these groups. So what we can see on the left is that there are a few populations, or a couple at the top there, again, which I've mentioned before, the CD8s, and also some Treg memory. And you can see that they're favoring the 1 dose of PCV10 versus 2 doses of PCV10. So really it's shifting, so the pattern is shifting either a little bit to the right, whereas with PCV13 after 1 dose versus 2 doses, you tend to see it favors more towards the 2 doses. So a lot of these cells tend to be activated more so after 2 doses of PCV13, whereas some of them, or a smaller subset of them, tend to be activated or enhanced after 1 dose of PCV10. So that's quite an interesting thing to see. And then when we kind of look at this directly comparing the 2 vaccines, we see something kind of similar, where we're actually showing that on the left is actually just following 1 dose of either vaccine, and you see it actually favoring PCV10. A lot of those cells are on the right of that dotted line, but following 2 doses of either vaccine actually favors more for PCV13, and that kind of is consistent with some of the data that we saw before, particularly from trial 2, the carriage data, where we did see quite a strong effect with PCV10. So this is quite interesting, and so this is very early days. We actually just got this data in the last few weeks, so we're kind of trying to get through it all and make some sense of it. But that's kind of where we are at the moment. In terms of just some take-home points, the vaccines themselves are very potent. We know that. They generate really substantial amounts of cellular immunity across lots of different cell types. We know that for some of these cells and some of these markers that we're looking at, they do appear to be similar across whether, regardless of the vaccine or the number of doses, but there are some key differences. And I'll try to show you that briefly, either between the vaccine, PCV 1 dose, or PCV10 1 dose, or PCV13 2 doses, which we think does support some of our antibody-based data, and hopefully might support some of the mechanism driving some of the carriage outcomes, but that's still some time away. And more importantly, we've been able to identify some interesting new subsets of cells involved in the response following PCV. So I've just highlighted 3. CD8s. CD8s are usually viral infection. They really respond to viral infection. So it is quite unusual that we do see them in this context. But of course, it's believed that following a vaccination, of course, the bacteria is engulfed and does become intracellular, and that's when the CD8s might enhance that clearance. I've explained the memory Tregs in terms of homing to the mucosa, maybe controlling some homeostasis at the mucosal sites, and then unconventional T cells, which are kind of a new type of T cell. Not a lot is known in relation to pneumococcal bacteria, but particularly following vaccination. So these are kind of new areas of lines of research that we're kind of looking into at the moment. But of course, yes, analysis is definitely ongoing. We've got a lot of data, as you can imagine. I've only really shown you day 28. I should have said that at the time. It's just only day 28, so 1 month post-vaccination. We have all this data again for day 7. And interestingly, we do see some different subsets appearing at day 7. And so that's kind of interesting. And we want to kind of have a global picture of what's going on with these vaccines, with these doses over time. And what we haven't done yet is analyze the innate cell population. So of course, in the early phase of infection or in vaccination, you get a lot of innate cells being driven. And so that's a lot of data that we need to get through as well. Our plan's really also to develop some new assays to look at these responses, to hone in on the serotype specific nature of the response, so we can have a similar kind of outcome to our antibody-based data. That will make the correlation analyses and so forth a lot easier. And then, of course, as part of the overall objective of the trial is to explore these relationships from these cells that we've identified with whether they can protect or not, or seem to be associated with protection against carriage. And so we will greatly rely on the expertise of Yimbun for that. And so we hope we can develop some models where we can incorporate a lot of these data together to get an understanding of what's actually going on. So that's all for me. I'd just like to acknowledge quite a few people. So a lot of the guys from my team, I mentioned Jeremy, who's really been leading all the flow cytometry work, but the 2 students, one I can see here in the audience is Sadvika, has done a lot of the work that I presented as part of her honours work, and Grace was the honours student last year who did a lot of the day 7 data. Of course, a lot of the team members who are here as well, and Beth and Kim have kind of been leading a lot of these trials. And of course, our friends and colleagues at the Pasteur Institute, Tuan and Tan, are here. We couldn't have done it without you. So it's been a great exercise, and we hope that there'll be lots more collaboration in the future. So with that, I'll leave it there, and yeah, thanks very much.
Thank you, Paul. So it's very interesting section this afternoon. We walk through from statistical approach to very complex clinical trial design, and also to understand about the use of high throughput flow cytometry to discover the new immunological marker to understand the immunogenicity of PCV vaccine. So now it's time for a question. Yeah. Anyone have a question? You? Okay.
I have a question for you because it's very interesting when we're talking about correlate protection. And because I work in clinical trial, and for me, RCT is the top of the design, study design. But now I hear about the post baseline. Something happened after we randomize. So my question is how we can handle it at the beginning, and any signal we can recognize this during the trial by interim data analysis.
So, Can you hear me? So thank you for the question. If I'm not mistaken, you are talking about that despite randomization, which is good data, despite randomization, things can happen after the randomization that can generate a problem for getting a valid signal and how to deal with that problem. Is that the question? Okay, so this is actually a quite a general issue, not only in the studies of COP, but also generally a problem in the studies in clinical trials. So clinical trials only guarantee comparability at baseline, but not subsequently. So what happens subsequently? What happens subsequently becomes messing up the randomization and therefore comparability. So basically, there are two things that we can try to do. One is, in a general sense, to enhance compliance so that there are less deviation from the randomized result to happen. So that is one aspect. That is the implementation of the study design. And another angle of this in the COP context would be, as I suggested just now, try to find a trial situation such as to find a seasonal transmission site such that we can do it earlier and prevent the post-randomization problem to happen. So that is one aspect. Another aspect is statistical. So in the last 10 years or so, there has been a quite substantial statistical methodological development to specifically deal with the adjustment of post-randomization variables. So that's a specific class of causal analysis that deals with what happens after the randomization, and if you need to conduct statistical analysis with adjustment of this post-randomization phenomena. So the technology is already there. Just that. So I got my PhD in year 2000, and for people like me, probably we are not learning a lot of things that are published in the last 10 years. So I think it is important that we keep updating. And as far as I can see, we already have the technology that has been developed in the last 10 years from the causal analysis strategy that we can deal with adjustment for randomization problem so that we can still get a signal.
Okay, thank you.
Thank you. We also have a question from Amaran Lish for Tupo. So why would this PCV10 1 versus PCV13 two doses happens? Serotypes or carrier protein differences? ICDC future works. Thank you.
Is it on? Yeah. Thanks, Amanda, again. Can anyone hear me? Is it working? Yeah, okay. Yeah, that's an interesting question. It's something that we are trying to work out why that would be. The obvious thing is carrier protein. We know there are differences in the carrier protein. I think, you know, it is a real result because we're seeing it across a number of different assays and studies, this PCV10 effect. I think one option would be, this one there. So one option would be that we would try to develop some of those serotype specific assays where we might be able to tease out those serotypes that are conjugated to different carrier proteins. Another option would be, and I guess study, Amanda, would be with Pneumococcal. Do we see something similar with Pneumococcal, for example?
North Building.
Yep. So that would be my thoughts at this stage. But yeah, definitely food for thought. Thanks.
Thanks, Paul. So hopefully, Amanda, you're happy with the answer. Any else have a question? Yes. Fraser, Shishida.
So Paul, I have a question for Paul. Very interesting immunological data, but as you know, pneumococcal carriage is very common. And what we are looking at the effect of vaccine response. How would you consider the natural exposure during this vaccine trial that happened. Because we cannot exclude that even before the child received the vaccine, the child may be exposed to naturally circulating pneumococcal. So they may have some minimal level of antibody or memory cell already in this population that we are investigating. And how would you consider that in the data analysis after 2 dose or 3 dose? That's one question. Another question is, I noticed that when Kim did a complicated trial in Ho Chi Minh, even 2 dose schedules, the second dose were giving like in 6 months and collecting the blood at 9 months, while in the 2 months, 4 months schedule, collecting the blood at 6 months. So there is an interval between 2 and 3 months, change it after the 2 dose schedule. And then when we assume that usually the primary vaccination is if the child received the vaccine for the 6 months of age, we usually consider as a primary series. And after 7 months or 9 months, we consider as a booster. But giving a 2 dose at 6 months and 2 months, whether that's a primary or booster is another thing I think we need to consider. So I want to know your insight on that issue.
Thank you. Yeah, I mean, I think maybe the second question first, the 2 dose interval. Yeah, we kind of we decided to call that as a 2 dose schedule rather than because the interval wasn't great enough to as a booster as such. So I think that's certainly there's age related effects, of course, but the response is very different, and that's something that you probably expect with those changes in interval. And we try to take that into account when we do the analysis. In terms of the other question around carriage exposure, yes, I mean, obviously that is a key factor, and we will, we have quite detailed carriage data from the guys sitting in the room here, so we'll be able to take that into account. Our responses are, immediately after vaccination. So, we do have baseline response, and we also have post-vaccination, and then we will have some longer-term data as well. So I think the challenge will be to try and find the right models that will incorporate the exposure, because I think the exposure in between the pre and the post is quite small. The window is very small, but it's what happens, in that interval. So, yeah, they're definitely good points, Lei. Thank you.
Okay.
Thank you, Paul. You have another question. It's from Chris Bly. So to extend Amanda's question to Paul, could mixed schedule enhance the T-cell impacts? And does your data give a strong indication on the order?
Another good question, Amanda.
From Chris.
Who's?
From Chris Bly.
Oh, Chris Bly. Sorry, Chris. Yes, that's an excellent question. Actually, we've been talking about this just recently, and Beth was the one that raised this. Could we have a schedule? And I know Amanda Leach has run this trial with combo, so cells would be quite interesting to look at in that situation. But yeah, we could envisage a situation where you might have a mixed schedule where you have PCV10 followed by PCV13, or in some form. And I guess the interval or the when those doses would be given is the question. But that's definitely something in our sort of sights. Thanks.
Thanks, Paul. Anyone have a question? I have one question for Dr. Tuan. So just would like to ask, you are consultant for the IPV in Vietnam. So based on your current data, I know that Vietnam plan to introduce PCV in next 3 years. So which schedule will you recommend for Vietnam?
I just work in the Boston Institute for Vaccine Clinical Trial, and I participate in some meeting. But for me, I think to put any new vaccine in the NIP, the question is, do we have enough data, and do we have the budget mechanism? So based on the current data we have, the reduced dose, and then the one that we have from Paul study, I think the schedule 1+1 or 2+1 is the best schedule for the low middle income country, because not only for the long-term protection, especially for the second year, but also they can put in the schedule of the EPI program. So that's my comment.
Thank you. Okay. So if no more questions, so then I would like to close this section, and we will have a next one.
Yes. Yeah. So thank you for your attention, and I just have a couple of housekeeping things. Yes. More.
Just one notice about today is release have a thunderstorm of asthma. So you may know for people not living in Melbourne. So Melbourne is the city with the high risk of allergy, and today is very high risk for people having asthma with the pollen index very high. So if you have underlying disease, so just be careful wearing masks outside or limiting your time outside.
Thank you, Anna. And I'm on strict instruction from Darren, otherwise I will cop it. So straight after this, there's no afternoon tea, but there are some breakout sessions, I think. So those of you that are participating in that, they will be held straight after this. So there's a microbiology session that you can probably talk to Catherine, I suppose. That seems in the Walford room. Is that correct, Catherine? Yep. And there's a health economics one in the Cox room. So Natalie's the one to look for that. They'll be straight after this. And for those of us who will be attending the dinner tonight, 6 o'clock MCRI reception, we can all meet down there unless you let Darren know that you're going to meet directly at the venue. So thanks everyone for your attention, and session's closed, and look forward to seeing those of you tonight.